In the previous video, we talked about how cells make proteins. It should be really obvious to you that it's an extremely important job for a cell to be able to make proteins. You learned about proteins in previous chapters. They provide support, movement, transport, protection, serve as enzymes, cell recognition, channels, carriers. Proteins are amazing, right? Well, cells also have to be able to make new cells. So cells divide in two ways depending on the type of cell. Mitosis occurs in what we call somatic cells. Meiosis will occur in the sex cells that allow us to make sperm and egg cells. Okay, so we'll talk more about that later. Let's focus on mitosis right now. So somatic cell division is when one cell divides and results in two genetically identical daughter cells. Now we do this to create the millions of cells or billions of cells in our body. So we're constantly having to create new cells to grow, to maintain, and repair. If you really want to see the need for new cells, vacuum your mattress. Yeah, pretty gross, right? Take your sheets off. Take your vacuum cleaner. Clean out the tank if you have the plastic one that you can see in, Clean out the tank and then take the hose and very slowly vacuum your mattress, especially the area where you sleep the most. Oh, the longer you've had that mattress, the more grossed out you're going to be when you see all that fluffy, brownish, gray gunk that's filling up your vacuum cleaner. Well, guys, most of that, dead skin cells. The cells on the outside of your skin are so far away from the nutrients in your blood supply, they're dead. So they're constantly sloughing off. Yeah, you'll start vacuuming your mattress now, won't you? Anyway, imagine what would happen to us if we weren't constantly making new cells. Yeah, we'd run out of skin. We wouldn't have any, oh man. When we start talking about tissues in the upcoming chapter, you'll understand how important this is. So we have to make new cells to build new tissues, to maintain, repair, and to grow. So the cell cycle is actually divided into two different phases. Interphase, which lasts about 23 hours, and mitosis, which the whole process takes about an hour. So cell cycle, think of it like the life cycle. What are you doing at this particular stage of your life cycle? Well, you may be raising children and going to school and working a job. Well, you were doing something different when you were a child. You weren't getting ready to reproduce and make children because you were a child, right? You were doing your child job. So think of interphase as the cell doing the cell job, and mitosis is the phase where new cells are made. We'll examine each of these stages individually here in your lecture, but you'll also get this in your lab as you learn how to identify these stages using the microscopes. So interphase is the time when the cell is getting ready to divide. The cell is doing its job. A liver cell is producing proteins. A toenail cell is producing toenail proteins. We subdivide this into three substages of interphase: G1, S, and G2. Now, it's important to remember that we're not yet dividing or replicating our DNA. So the DNA is in that loose chromatin stage, just sort of wadded up, stringy DNA, okay? So the first part of interphase we call G1 for growth or gap 1. And this is where the cells are growing and producing new organelles and the new structures that are needed for DNA replication, which happens in the S or synthesis phase. So the double helix strands of DNA are going to be replicated. We're going to look at DNA replication, so I'm not going to elaborate on that until we get to the next slide. But the third subphase of interphase is G2, gap or growth 2. And it's during this phase where the centriole replication has been completed. So we've got two centriole pairs that are going to be there in the cell. We're making the enzymes and the other structures that are needed for mitosis or cell division to occur. For DNA to replicate, or basically make a copy of itself, the first thing that it has to do is separate again. So DNA helicates breaks the bonds that hold the two strands of DNA together. That causes the double helix to uncoil. Then polymerase, remember, polymerase, make a polymer. Polymerase is going to add base pairs. Now, these are all found within the nucleus. It's sort of like a pantry. We need to go get the stuff we need to make what we need to make. So we're making DNA. We're replicating DNA. So if we unwind both strands, or unwind the two strands from each other, then we're going to add base pairs to both strands. So if we've got a strand of A's, T's, C's, and G's up here, and a strand of A's, T's, C's, and G's up here, and we add more A's, T's, C's, and G's to complementary base pair, we end up with two identical strands of DNA. Now, if you don't believe me, try it yourself. Write out, make yourself a strand of A's, T's, C's, and G's, and then complementary base pair them. Then break them apart. And then what do? You add A's, T's, C's, and G's to each one, and bam, you end up with two genetically identical strands of DNA. All right. So enzymes are going to proofread this for errors to make sure that everything was replicated. And we call this semi-conservative replication because each new strand of DNA that we just made means that each nucleus of the two new cells we're going to make will have one strand of the old DNA and one strand of the new DNA that we're adding those bases and making. Now, as we just said, during interphase, the cell is doing the cell job, and when it's getting ready to divide, it grows, it creates new DNA, new cell membrane, new organelles, everything that you would need to separate one cell into two cells. So mitosis is that time of nuclear division. Divide the contents of the nucleus. Now remember, back up in the S phase, we doubled the DNA, right? So instead of having 46 chromosomes, now we got two copies of 46 chromosomes. So mitosis will separate the contents of the nucleus. Cytokinesis separates the cell. So it separates the cytoplasm and the organelles, and one cell becomes two. When we look at mitosis, we divide it into four consecutive distinctive phases: prophase, metaphase, anaphase, and telophase, or PMAT. Or if you want to throw the interphase in there, it would be IPMAT. See? All right, let's look at each one of these individually. Prophase is the first stage of mitosis. Now remember, during interphase, all of the DNA was jumbled up in chromatin, right? Just long, stringy mass. Well, in prophase, that DNA is going to wrap and coil and twist around all of those histone proteins that are associated with the chromatin. So they coil around and they become chromosomes that are composed of two sister chromatids. They're just sort of short, thick rods. Chromosomes become noticeable during prophase when you look at them under a microscope because they are more structured rather than just being all stringy masses, right? Now, when you look at this under the microscope, as you can see right here, you're not going to see little X-shaped chromosomes, but you can see in the cartoony image that we have created two copies, and those are your sister chromatids, and when we stain them, they do become visible. The other major thing that happens during prophase is the nuclear envelope dissolves. Remember, the nuclear envelope is like the cell membrane, but it's just around the nucleus. So if we're going to replicate all this DNA and then separate it into two cells, we can't do that if it's still housed in a container. So the nuclear envelope dissolves, the centrioles are going to start to grow spindle fibers. We'll talk about those in just a second. And they're going to be pushed to the opposite parts of the cell. And we call that the opposite poles. So to two opposite sides. Metaphase is the next phase. The chromosomes will get pushed toward the middle of the cell as the spindle fibers continue to elongate. Now the spindle fibers are also going to attach to the centromere that holds those two sister chromatids together. So you can look at an image of that here, or a cartoony image. So here are your centrosomes, there's your chromosomes, your spindle fibers attached to those. Now that causes all of the chromosomes to align at the metaphase plate, which is in the center of the cell. The third stage is anaphase. It follows the metaphase. The centromeres split, and the spindle fibers pull the sister chromatids apart. Now, I think of this like fishing. So if you think of this, your centromeres, your centrosomes up here as being your fishing rod, then the spindle fibers are your fishing line. And when they attach to the chromosomes, you've caught a fish. Now, all of these catch a fish at once, so we start reeling it in on each side, and what does that do? It pulls those sister chromatids apart as we reel them in. Now, what we're really doing is separating those two copies of the DNA so that we can put each copy into a daughter cell. And after we pull those chromosomes apart in anaphase, telophase is going to continue to separate those cells. So the chromosomes are going to decondense back into chromatin. The nucleolus and the nucleus, or the nuclear envelope, begin to reform around that DNA. The spindle fibers are broken down so that they can be recycled. And when you're looking at this in the lab, you look for this structure right here that is called a cleavage furrow. So this forms as there are contractile proteins being created that are squeezing all of this together, and that is going to lead to cytokinesis. So cytokinesis is when the cytoplasm completely separates, a new cell membrane forms and closes off, and now we have two completely separate, genetically identical cells that go right back into interphase and do the job of the cell that started this whole process, because it is genetically identical. It does the same thing. So if the cell we started with was a liver cell making clotting proteins, now we have two new cells in the liver making clotting proteins. Aging is a normal process that starts as soon as you're born, and guess what, guys? It keeps on until you die. Now, there are some things about aging that are very easy to see and very obvious. Wrinkles, decreased muscle tone. So you can look at someone that is 90 and see that their body looks very different from someone that is 9. One of the things that happen is atrophy. So cell death leads to a decrease in the size and the function of an organ or a tissue. Hyperplasia is overgrowth. So as we accumulate DNA mutations throughout our lifespan, sometimes that can lead to hyperplasia or overgrowth, uncontrolled mitosis. We call that cancer, and it results in tumors. There are other types of hyperplasia that are not cancerous, such as benign hyperplasia of the prostate gland. Prostate just grows and interferes with normal function. So how do cells die? Well, they do so in one of two ways. They're either harmed by some type of damage, whether it may be chemicals or it may just be you got an injury, or they commit suicide. We call that apoptosis or autolysis. Auto means self. Apoptosis is known as cell death. So it occurs normally in order to get rid of cells that are worn out so that we can replace them with new cells. It happens when we have too many cells. Cells are old. We have bad or damaged cells. But it's also a part of our development. So when we are early during our embryonic and fetal development stages, we have this webbing that's between our fingers and toes. Well, we don't have that when we're born. So some of those cells will die due to apoptosis or autolysis, so that we're not born with that extra tissue. So it just helps us to form normally. However, this can be utilized with chemotherapy as well to target cells that are part of tumors, so that we can destroy them by initiating apoptosis. All right, guys, congratulations. You made it through chapter 4. You should be getting ready for your first exam now.